Hello. So we were talking about what are unculturable bacteria, right? So when it comes to unculturable bacteria, what is by definition they are? What are the reasons why are they not growing in the laboratory? We have been discussing about it. So we all know that the simple explanation why these bacteria are not growing in the laboratory, why are they unculturable? Why are the microbiologists failing to replicate these bacteria in the laboratory? They are existing in the environment, but they are not uh, growing into the laboratory under laboratory conditions, and why this is happening. So this remain the topic of our discussion for last entire week, and we continue with that today also. So of course, it is not that we don't know the methods to trap those organisms and grow them. Different aspects of the environment are to be explored and hit the one which is right for uh, finding that organism of your interest, right? So we have seen that environment factors like nutrients, pH, osmotic conditions, temperature, and many more factors are there we need to be taken care of. So attempting to mimic various conditions which are there under natural conditions to be mimicked under laboratory condition is a challenge. It is a multidimensional matrix, you can say, right? You have n number of possibilities. You have to exhaustively address and find out which would be the right strategy to bring that organism grow it under laboratory. So you'll have to like prepare tailor-made synthetic media might be it is required so that the suspected environmental condition you are providing the organism and it finds it happy and conducive and starts growing. So, with various approaches to synthesize media to mimic the environmental conditions we have discussed in previous lectures. Now, I am thinking that shall I like summarize the different approaches which have been discussed for culturing the missing bacterial diversity, you can say the uncultured bacteria, they are missing as on record what we have today. They are not seen, they are not reported. So people have tried a number of methods, and out of which a few we have discussed. The summary of some of those we can rediscuss now again. All right. So when it comes to these bacteria, growing them in the laboratory, of course the very first is going to be mimicking the natural environment, right? So you can have a simulator environment. So of course it is difficult to replicate natural environment exactly by synthetic means. So can you just manage to bring that natural environment to your laboratory? That is a part of it you are going to not create but trap in the laboratory, right? So maybe the ocean water sample across its depth, right? You are going to collect and put it in an aquarium. That example to the details we have learned. So alternative to take the bacteria back to the environment to grow them, right? You are not totally taking them away from the conditions what they are dwelling in, because the entire water sample you have brought from the site where they are naturally growing, and just that you are putting it in an aquarium and trying to maintain the physical parameters under those conditions. That is one aspect. Now second that we had talked about was the diffusion chamber, right? You remember the diffusion chamber in which we have a steel plate and it has got semi-permeable membranes. Between the two membranes you are going to have the gel matrix onto which you are going to inoculate. So you are enclosing the bacteria within a semi-permeable membrane, and the cells are unable to pass through the membrane, right? Because the pore size matters. So you are selecting the filter such that, or semi-permeable membrane such that the bacteria will be retained. It will not pass through the membrane, but movement across the membrane, that of nutrients and growth factors from the environment is going to be allowed. So these chambers are sealed So you have the chamber, you have that gel, you have inoculated it with the sample taken, and it is sealed, right? So when you are going to inoculate, of course, dilute suspension of the cells, or dilute suspension of the source material, may it be from marine water or any freshwater sample or any degrading organic mass that you have collected from some plant deterioration site, right? Because you want to find out what are lignin and cellulose degrading bacteria. It depends on your intention to isolate, or if it is just to see the diversity of bacteria there. So you can have any sample of that kind, right? You are going to dilute it, and then you are going to inoculate into that chamber. The chamber is sealed, and then it is floated onto the support system, you can say. So for example, it is an aquarium in which you have mimicked the natural environmental condition, and on the top of that, like you put the paper boats, you have floated this sealed diffusion chamber, right? So liquid can pass across, but bacteria will not. So whatever is in growing within the sealed disk is going to remain uncontaminated from the source. It is not going to receive any bacteria, or no bacteria from the disk is going to move down into the aquarium, right? So these are the tight chambers. So slowly as the time passes, the small microcolonies will appear on the gel, and that you can take for observation under microscope. Of course, it is not that key once you have taken it out, you have exposed it to microscope, then of course is going to be a challenge to put it back, because you are exposing it to the laboratory conditions in the open, uncontrolled, non-safe, the open air, right? So aseptic conditions need to be maintained if you want to reuse that disk. That is very clear to us. So if you see the success or efficiency of this diffusion chamber as a technique to isolate unculturable bacteria, people have succeeded to get almost 40% of new isolates from the same source, and the diversity to 40% could be explored. Otherwise, hardly 0.05% from the source. So, for example, you are taking some soil sample and you are trying by normal laboratory isolation methods like enrichment, etc., that we have discussed earlier. You may tap those which are R type of strategies, which are growing very fast, and are not going to require any specific fast radius requirements. But other S type of strategies, they are going to get masked, how that we have learned earlier. So only 0.5% maximum, I'm talking about hardly 0.5% isolates you can tap by this method. But if you go with advanced approaches like say diffusion chamber in this case, or the trap that we had discussed earlier, again we will take the revision of that, can give you a better isolation, a better exploration of the diversity. Those which were uncultured earlier will appear in that. So the extension of diffusion chamber is the trap, right? So it is like the word itself is telling you it's a trap. So the membranes are going to enclose it, and the pore sizes are going to vary. So selectively to grow the small sized filamentous structures, like say from actinomycetes, right? So actinomycetes filaments, they will be growing, but the normal bacterial cells, they will not be growing. So you may be like thinking of what kind of pore size it could be. So filaments of say 0.03 micron, etc., that pore size you select. So in similar to your diffusion chamber, you can have the trap system. This is what we had talked about, you remember? So you have filters, but the lower filter is of smaller pore size. So if the bacteria are there, they are going to be of course bigger than 0.03 micron size, so they will be retained on the gel, that is encased in the disc, but the fibrils or filaments will be able to pass across the membrane because their dimensions are less than 0.03. So selectively for trapping the actinomycetes, this experiment people have done. You can go with the you can say advanced version of diffusion chamber where the membrane size is different. This is what we had talked in previous lectures, right? Now it is very clear to you that when you are talking about isolating the unculturable bacteria, the approaches are very important. So you are trying to bring the environment to the laboratory. That could be done. In this context, yesterday we had talked about an example of Prochlorococcus, right? And its dependence on Alteromonas. Is it Alteromonas? Who will tell me? Is it Alteromonas or not? Yes, it is Alteromonas. It is known to produce catalase enzyme, and which is a the methodology or technique to fight back the stress, oxidative stress in the environment. And Prochlorococcus has its HIL genes, which are going to be helpful for it to combat the oxidative stress. But under elevated CO2 conditions, both these mechanisms of both these bacteria, that of Prochlorococcus and Alteromonas, gets compromised, and oxidative stress builds up, and these bacteria are going to be killed. Now, this is the interaction between these two type of bacteria dwelling the same environment, right? This is what we had talked about. But very clear from this study, we come to know that the catalase produced by Alteromonas is going to be important as a oxidative stress management system in the seawater, marine water system or ocean system, where Prochlorococcus is the dominant photosynthetic cyanobacteria. And without such catalysts being produced by Alteromonas, oxidative stress is going to be a challenge for survival and propagation of Prochlorococcus, right? You remember this. Now, similarly, there is another example, that is Alphanoproteobacteria. I repeat the name, Alpha bacteria. So it's a widespread organism, free living bacteria which are commonly found in seawater, but they are also found in freshwater lakes. They are reported in freshwater as well as seawater environment. Now, initially they were considered to be ubiquitous, that is present everywhere. But when it came to culturing these organisms, it was an uncultured bacteria, right? So this proteorhodopsin is the pigment this bacteria is having, and it is going to be essential one for working of light driven proton pump, right, which is the mechanism in the photosynthetic bacteria in the marine environment. Now, the DNA sequence of this uncultured bacteria that was studied. And why was it studied? Because it is another example. Prochlorococcus is the one which is one of the abundant cyanobacteria, which is important in carbon fixation through photosynthesis. This is another example. We are talking about proteobacteria. The species is alpha proteobacteria, specifically been studied by the scientist in this particular paper which I am referring to. However, proteo bacteria, they have proteorhodopsin, and this they are the one which are of importance because the carbon and energy balance of the ocean is important, and these bacteria, they are the game players. Yeah, right. So I'm referring to the paper by Giovannoni et al., who had studied these bacteria and their variants to the details. They have attempted to isolate it. So they had taken the environmental sample, and the most abundant organism they wanted to grow, but it wasn't growing in the laboratory, right? So they had tried to use the microtiter plates as the culture plates to grow them in the pure culture. And what they had given the name to this bacteria is Candidatus Pelagibacter. Candidatus Pelagibacter ubique. That is what the name given them to these organisms, group of organisms, you can say. They were isolated from marine water source. But uh, the story is that they have to make an attempt to grow them in n number of ways. Isolating their DNA from the sample was easy, and they had done that. But when it comes to culturing, it was a challenge again. But of course, now it is been explored and grown into the under laboratory conditions. All right. So I'm sharing the slide with you so that you get to read through, right? And understand what I'm talking about. So this is a kind of aquarium, the mini system that they had constructed in the laboratory, and it is mimicking everything, right, from the bed of the aquarium to its top. It is very much like a fraction of or section of the ocean water, right? You have all the sediment materials, the pebbles, the sands, and all flora, fauna, etc. So the SAR 11, that is the isolate name they had given, that of alpha proteobacteria, uh, th- they could grow it with the help of such kind of small aquarium that was established into the laboratory, and then using diffusion chamber, they could they could find these organisms growing. I mean, they could grow it as a pure culture. Now, when it comes to prochlorococcus, or maybe it is alpha proteobacter, something which is provided by their environment they are requiring, and which where we fail to understand that we can't grow them. All right. So a similar example we are going to discuss here, that of prochlorococcus again, but the aspect is different. So here is the aspect dependent strain. So you have prochlorococcus and a dependent strain of prochlorococcus. The name of the strain given is MIT 9215. Now, what is the difference? You have the susceptibility towards streptomycin is different. So you have mutant of prochlorococcus, which is streptomycin resistant mutant. Now, these mutants, they are abundant when you are talking about the natural source. You are taking it to the laboratory and trying to see these resistant bacteria are abundantly present around. And unless these bacteria are present, the streptomycin sensitive prochlorococcus growing or separating it from these resistant bacteria was a challenge. So. Uh, if you add streptomycin to the media, the sensitive isolates will get killed, but only the resistant ones they will going to survive. But the point is, um, kill the helper population by treatment with streptomycin will be possible. But as the helper population is killed, the streptomycin resistant bacteria, which is resistant to streptomycin, growing on streptomycin media, it also will consequently get killed. How it will happen? The term itself says the streptomycin sensitive strain is the helper strain, right? Because of its presence, the streptomycin resistant prochlorococcus could grow. So maintaining the culture at high density, of course, minimal dilution or subculture. You are aware of these terminologies, right? Minimal dilution. So dilution may you go on reducing the number of cells per unit volume, and in subculture, you have to assure that the inoculum size is appropriate so that you get well isolated colonies. At the same time, the growth is good. So in pure cultures, these organisms, when inoculated at high density, they could propagate in pure cultures. But the concentration of prochlorococcus cells, when they were diluted below about 10 raised to 5 cells per ml, the culture failed to grow. So, and if you want to have the isolated colonies of these prochlorococcus MIT 9215, it was really very difficult to find them growing on the Petri plate as isolated colonies. Right. So large number of different helper bacteria that allow the growth of these prochlorococcus strain are there, and that is what people have researched on. So they could find that yes, like in case of prochlorococcus and alteromonas, we have seen the catalyst requirement is the dependence factor, and without that catalyst produced by the helper bacteria, alteromonas, right? The helper bacteria is Alteromonas. Prochlorococcus cannot survive longer under high CO2 conditions. So similarly, here they had attempted to add catalase, the enzyme that breaks the oxidative stress. Hydrogen peroxide is broken down. We know that. So that helped the growth of these bacteria, MIT 9215, which is streptomycin resistant on plates. But removal of H2O2, as we know, it is very important to reduce the oxidative stress, right? And for it is going to help the growth of these bacteria because oxidative stress is removed. So natural level of H2O2 in surface water, if you see, is high enough. So it has to take help of those bacteria which are producing catalase naturally. So among Prochlorococcus, you have two types: the streptomycin-resistant mutant, which is dependent strain of Prochlorococcus. So it is just to make you understand that bacteria in their natural environments are so closely interacting with each other and are interdependent that mimicking the environment that actually they are living in, uh, to be mimicked under laboratory conditions is a big challenge. All right. Now we go to the next concept, that is crowded isolation plate technique. Here we are taking an example of Marybacterium polysiphony. Marybacterium polysiphony is the one. It is again once was uncultured bacteria, and it was chosen as a model to identify the mechanism of codependence. The new term like catalase by Alteromonas and Prochlorococcus, again it's a kind of codependence, right? But it is one sided. So here it is codependence we are talking about, with another example. Now, this Marybacter was chosen as a model system to understand this codependence. Now, here, uh, what they found when the sample was taken from natural environment, these bacteria were able to grow in liquid cultures where other bacteria were present in the solution. But when they were tried to put on the Petri plate, they could not grow. But what now can be done? By then the scientist had understood of I'm sorry, by then the scientist had understood the mechanism of interdependence and under natural environment. So they had done certain experiments, which of these bacteria would help this Marybacter to grow under laboratory conditions on Petri plate, like Alteromonas helps Prochlorococcus on similar line. And surprisingly, what they found, one strain of E. coli helped the Marybacter polysiphone KLE1104 to grow. Now, what they did, why this particular strain only, or any other any other strain of E. coli also would help. So they had developed the mutants of that E. coli and had checked whether these mutants also are helping growth of Marybacterium. So what actually happened when they created the mutants, that some of the genes, they were knocked out, they were silenced, right? And the mutant with the silenced gene was not able to produce enterobactin, right? So enterobactin synthesis pathway was enabled mm, due to these mutations. Now that bacteria was inoculated for as a co-culture with Marybacterium. And what happened? The bacterial strain, which had the enterobacter synthesis pathway genes knocked out, it couldn't help the KLE1104 to grow. That means there is something which is from the enterobacter synthesis pathway that is helping the KLE1104, that is Marybacterium. Yes, enterobactin itself is a siderophore, right? So that siderophore requirement uh, is the necessary factor when it comes to growing Marybacter under laboratory conditions, right? So we can just see the experiment, the steps of the experiment, so that uh, how they must have elucidated the fact that enterobacterium is required, and it is provided by the E. coli which is growing in the medium, right? Now They had collected the environmental sample, and it was spreaded onto the petri plate. What happened, as it was the entire sample, a few hundred colonies had grown, right? That is quite expected. Now, uh, they were interested in isolating that maribacterium. So every time they are studying a colony, they are trying to identify it by various mechanisms and trying to find whether that is maribacterium. So in this studies, what they had done is they had selected the paired colonies, right? And the paired colonies were picked up and they were cross strict on the petri plates. Can you imagine what how it would look like if it is cross strict on the petri plate? So in one corner you are inoculating one isolate, in the other corner you are inoculating the other isolate, and then you are spreading them to cross each other's growth, right? So cross trick, that is like four quadrant trick you do, that is for one bacterium. Here you are going to use two different isolates, but which ones? Which are growing as pairs in the in the petri plate, right? Those two isolates are selected, and then they are inoculated as cross tricks, right? Now because they are cross tricked, there are, what are the possibilities? The possibilities are that if they are going to be helpful, growth of both will be taking place. If they are not really helpful for each other's growth, growth of one will take, another will get inhibited. That is quite possible, right? So they had tried all the different colony pairs, that those were cross-tricked and were screened for dependence of growth, right? Whether the one bacteria that is their interest, the bacteria of their interest, Meribacter, is dependent on which of the others which are obtained from the environment is to be tapped. So you have n number of cultures growing into that petri plate, and you have to find out which among those is the helper for Meribacteria. So they have selected the paired colonies and they have cross-tricked and seen which of them is helping the growth of Meribacteria. It was found to be E. coli, and that E. coli strain was able to produce enterobactin. Enterobactin is a siderophore. So siderophore codependence uh, is concluded while growing this Maribacter Polycephonie Kaylee 1104. So this was another example when we talk about isolating the once uncultured bacteria, right? So in short, when you are talking about uncultured bacteria, which are going to grow in under conditions which are very similar to their natural environment. That means they are familiar environment you are providing to them. So they are going to recognize the presence of certain growth factors which are released by their neighbors, right? And we cannot imagine what is its neighbor and what kind of a growth factor is released by his neighbor. So we have to have such mini aquaria or mini miniatures of the natural system to be established in the laboratory and go on experimenting. So when we talk about co-culture of organisms from marine sediments, the diffusible molecules, right, like that of siderophores, are available. The siderophores are produced by the neighboring species, and they are released into the media and are going to help the growth of bacteria of your interest. What are the siderophores? These are the low molecular weight compounds, we know, and they have you can say specific affinity for insoluble Fe3 plus. So microorganisms release siderophores. What must be the function? It is to make the ion available, right? Fe ions available. So siderophores are released. Enterobacterium is released to scavenge Fe3, and then it will be transported to the cells, right? So ferric ions uh, availability is assured through siderophores, right? So soluble Fe2 ions, if you talk about, it is limited in most of the aerobic environments. So siderophore biosynthesis is going to be an important mechanism. Of course, it is tightly regulated in culture species. Under natural environment, the story is different, and when you're trying to grow them under laboratory conditions, there are different challenges. So, growth of siderophore producing bacteria and production of siderophore or biosynthesis of siderophore by these species is a challenge. The chelators, like siderophore molecules, are the chelators, right? They are going to chelate Fe ions. They will be produced when the available iron is low. So you have to design the media in such a way that it is going to provide very less amount of iron, right? So uncultured species would be expected to have the same ability to produce siderophores or to scavenge the siderophores um, uh, uh, from the environment, right? So uncultured species, if you are talking about those which have the ability to produce siderophores, and they have their genes functional in the natural environment because iron availability is very low, and but when you are putting them under artificial conditions, your nutrient combination that you are providing is making iron available to them, and siderophore production won't be there. So while exploring co-cultivation or co-dependence culture methods, also you have to be aware of what environmental parameters you should provide to the cells which are going to act as helper cells, right? Now, the experiment that we discussed told you that this Kali4 is going to require the siderophores which are produced and provided by mm, E. coli isolates. But what happened to that E. coli if its enterobacterian producing capacity is compromised by inducing mutations? Then that siderophores are not produced, and then it will not act as a helper bacteria. All right. So enterobacterian, of course, as I said earlier, is a siderophore, the secretory molecules which are going to solubilize the oxidized iron Fe3, and then this iron is made available as a nutrient to the cells. So spent medium, that is where earlier you have grown that E. coli and then remove the cells, and the remaining media, if you can uh, take it as the supplement, right? So you want to grow your micrococcus luteus is another example. So this micrococcus you want to grow, which is again another helper medium, helper organism. So in the medium, either of these bacteria you will grow, you will take the culture filtrate, which is called as the spent medium, and this medium you will add to the culture media of the bacteria that was once uncultured, and you want to grow it with the help of some secretory molecules that you have added in the form of spent medium. So this is another concept, that is helper siderophores. It is one of the concepts. All right. So if I say, do bacteria produce it under always produce such molecules always? No, again it is the environment that is going to select expression of genes responsible for siderophore production. As I said, one of the conditions is it is to be very deficient in iron availability. If it is very less in iron content, then and then only bacteria will not get it that easily, and it will have to make efforts to find it. So siderophore is a kind of effort made by those bacterial strains to have the capacity to produce it and scavenge that Fe3 which is there in the environment, and Fe3 converted to Fe2, and so on and so forth. All right. Now here the second part of the story, as you are seeing 6 plates here, right? So this is KLE1104 Marybacterium, which you want to grow because it is the uncultured species, and you are trying to grow it in the laboratory, and now you are uh, concluded that yes, E. coli enterobacterium producing E. coli strain is acting as helper. Now what happened is the mutations that those were introduced, and those bacteria E. coli strains, they were grown in different vessels, so different mutants in different separate culture vessels. And after the growth of those E. coli and their mutants, each culture vessel was harvested, and the spent medium was used to grow Marybacterium. Now here in this slide you can see the effects demonstrated by different mutants. For example, in this case, do you see the culture filtrate is there in the disk, right? So there are different ways to add the culture filtrate. Maybe you are going to put a liquid medium. In the liquid medium, you add the spent medium. On the top of that, you put the filter paper, and on that you inoculate the organism of your interest, that is Maribacterium. Or maybe like this diffusion assay that you do for antibiotic assay or vitamin assay, right? So the medium is there. You are going to bore a well. Into that, you are going to put the spent medium, and you have inoculated your the sample source where the suspected bacteria, or I should say desired bacteria, is present. So I have put it. Now I don't see any growth here, right? But I see a growth which encircles the well in which I had put the spent medium. Same is here. I am seeing no growth around anywhere else, but it is just surrounding the well in which I had put the spent medium from. Now, which is the spent medium, source of the spent medium? Here it is E. coli WT, here it is the E. coli Delta Lux S. These are the different strains, right? So the variants of E. coli. But here in the third case, again you are seeing the diffusion of this nutrient which is there, that spent medium that was put into the well has allowed the growth, and the growth is really well, right? But as against this, if you talk about example like delta ENTC and that of delta ENTB, there is no growth at all that of Meribacter, right? So what it is telling you, ki these mutants have lost the capacity to produce the growth factor that was helping Meribacter to grow. So these experiments were done to find out which is the growth factor required. So they had silenced different genes or pathways which are responsible for production of various secretory molecules. E. coli is producing various secretory molecules. So each type of secretory molecule has its own pathway for synthesis and the genes for its synthesis. So they had experiment by experiment, they had silent one type of gene set and one pathway, and stopped the secretion of one particular secretory molecule, and named it as say for example delta lux S or delta TNA or delta ENTB. These are the names based on the pathway they had silenced. Now if it is silenced, then there is no expression, right? That means it is the molecule which is required, and is because it is silenced, it is not produced, it is not available, like that. So what they had observed is the growth induction of a Maribacterium, that KLE1104 strain, by enterobactin, and enterobactin produced by E. coli. So for which they had used filter spent supernatants from E. coli isolate number BW. The number is not really important, it is specific to that lab conditions. But concern is enterobacterium, and enterobacterium is produced by that particular strain is a siderophore containing secretion, and so it is helping the growth of Maribacterium. So here spent culture media from enterobacter mutant strains, they do not allow the growth of Maribacter. So supernatant from the parental E. coli, that is BW25113, induces growth, but the strains which are deficient in specific genes and sequences which are required for